Question number 18 is Crescent of Giannuzzi is a microscopic feature seen in parotid salivary glands, submandibular salivary glands, sublingual salivary gland, or the lingual salivary glands. Now, to understand this, you need to know the histology of salivary glands, the large salivary glands, parotid, submandibular, sublingual, right? So, they are basically like compound tubuloacinar glands. They have ducts. That duct actually then diverges and ramifies into several, just like you remember the pattern of this bronchial tree, bronchial apparatus. Similarly, is the pattern here also. So they keep on dividing like this to form different tubules. It's like this. This is how a salivary gland is being formed. The duct actually branch and rebranch like this. So this is how it all ramifies like this. Anyways, so at the ends of these you find is like the acini, and these acini, it may be mucus secreting acini or maybe serous secreting acini. Well, I need not draw all of them. So what we're concerned about here is talking about the acini. So let's say that this is an acini of a mucous salivary gland, and this will be an acini of a serous salivary gland. So difference is that in the mucous acini you find is the basal nucleus and larger cells with the mucus secreting acini towards the apices, but those will not take good stain, because they are neutral substances. Unlike to this serous secreting acini, they have a central nucleus and they will have the protein synthesizing apparatus, that is the rough endoplasmic reticulum, ribosomes. They will all be basal to the nucleus. What will be towards the apices of these cells, towards the apical surface, they will be vesicles containing zymogen granules. So because all these proteinaceous structures, these serous containing acini, they take good stain, the violet stain. So in a histological slide, if it's a serous salivary gland, that will be darkly stained with a purple or violet dye. In case of mucous acini, you will see pale, you won't find a good stain there, like in the case of goblet cells. Now, in case of mixed salivary gland, not all, but some acini you will find is like this. You have this mucus secreting acini here, and this mucus secreting acini, I've already told you, it's like this with the basal nuclei, but there will be some acini which will override to this, like this. This acini is overriding to this acini like this. And this is a serous acini. Components of the serous acini, I have told you, you can find a nucleus there in all of them, right? They will be taking good of stain. So this is how it is. Now if you take a transverse section of this acini, you will find it appears like this. A central acini with large cells which are actually mucus secreting cells, but it will be surrounded by another acini like this. Such an acini surrounding an acini in a crescentic way, because in a section you find is like a crescent. Mucus, of course, will be drained out like this. And what about the secretions of this serous acini? Serous secretions actually are drained through this intercellular canaliculi of this mucus cells, and then it also drains through the same ductules. That is the pattern of drainage. So remember that the serous acini encapsulate the mucus secreting acini in case of mixed type of salivary glands. So this is a feature of mixed salivary gland, and this you will find in submandibular salivary gland. Mainly serous salivary gland, the example is parotid, and mainly mucous salivary gland, example is sublingual salivary glands. These crescents, they are called Crescent of Giannuzzi, or they're also called Demilune of Heidenhain. They're a feature of mixed salivary gland, and that example of mixed salivary gland is submandibular salivary gland. So the answer here is Crescent of Giannuzzi. Answer will be submandibular salivary glands.